Hi. So, in previous several sessions you have learnt about the innate immunity, and now I will discuss how this innate immunity knowledge or the information which we gained in last about 3 decades can be translated into therapeutic. Okay. And just for your information, the philosophy behind the disease or the concept behind the disease. So, any disease basically happen in the host by some perturbation by external environment. External environment, when I say, it means it could be environmental factor, it could be some pathogenic challenge or some other factors, right, physical or chemical factors. Okay. So, these factors basically perturb the homeostasis. Okay. And when this homeostasis is perturbed, then that result to the development of disease. In a very simple in words, how I can explain that, if there is some hypo situation or hyper situation. Hypo situation means it could be anything. Okay. Hypo means under activation of signaling pathway, under production of some molecules which is produced by this particular signaling pathway, under nutrition or malnutrition. So, all those things can result to the development of disease. On another hand, if it is too much activated, if any production of molecule or any signaling pathway is too much activated, then that result to the hyperactivation. Over nutrition also result to the development of metabolic diseases. Okay. So, the concept is, when this homeostatic range is disturbed by any mean, okay, then that result to the development of disease. Okay. For example, if you remember the previous session, I told that there is a LPS, and this LPS is sensed by the TLR4. If this ligand is too much, then there will be too much production of inflammatory cytokine. Okay. And if there is some mutation in this TLR gene which will not induce appropriately this TLR signaling, that is also a problem. Okay. If you remember, there is a disease associated with that is known as atrophy. Okay. So, overall the concept for the disease is, if there is a skewing of this homeostatic range in lower side or higher side, then that result to the development of disease. Okay. And in therapeutic, basically what we do, we basically try to manage this hypo response or hyper response by some agonist or antagonist. Okay. And that basically bring it to the homeostatic range. Okay. And those molecules, we call it as a therapeutic molecule, those procedure we call it as a therapeutic procedure, and all those things. Okay. So, let us begin with this TLR and therapeutic, and before that I will take you to a little history of immunotherapy. Okay. And this immunotherapy is basically started quite early, and this was started by one doctor. His name is William B. Coley. Okay. He was there, he was basically a cancer expert, okay, or most likely he is a oncosurgeon. Okay. He injected, initially I have learnt his little history. Initially, by accident, he observed the cancer patient who are infected by some bacterial infection. Okay. Those patients are recovered from the cancer, or they had less severity in cancer. Okay. So this observation was taken further by William Coley, okay, and then he intensely injected the streptococcal a cocktail of streptococcus, okay, in the cancer patient, in which the surgery is a little complicated or difficult, okay. And then what he found out that the individual who received this cocktail of bacteria, okay, their tumor was shrinked, okay, reduced, okay. In some cases this is completely disappear, okay, this tumorous growth, okay. So this observation was quite well accepted. This is a very interesting thing, that you put some, or you infect the individual with some bacteria, and that bacteria will recover from the cancer. That is very interesting. But unfortunately he was not able to explain this observation on the basis of science, okay, or logic at that time, because there was no knowledge of this innate immunity or PRR, PAMP and all those things, which was discovered in 1989, right. So this observation was quite interesting, and then this cocktail of this bacteria, he named it as a kind of coletoxins, okay. So coletoxin is a toxin which is consist of a cocktail of bacteria, and when it is injected in the tumor directly or in patient, then there is a recovery from the cancer, okay. And this therapy was very well practiced at that time, but since he was not able to give the scientific explanation, so he did not get too much recognition, okay. So Coley injected more than 1000 cancer patients with this bacteria and bacterial product, and many of these patients were recovered or reduced, their cancer was reduced, okay. And these bacteria or bacterial products are known as coletoxin, and are used in the treatment of bone and soft tissue sarcoma or cancer, okay. So here there is a picture, and why I am giving you this background, because there is a very strong basis of the scientific basis which he was not able to explain. Now we can understand this is basically the non-specific activation of innate immunity, okay. And this non-specific activation of innate immunity result to the activation of specific cells such as NK cell and other cells, and that basically cause the shrinkage of this cancerous tissue or elimination of these cancerous tissue. So now we can understand this thing very well, and therefore there is a various molecule, or there are various ligands of TLR which is used for the cancer therapy. Here you can see this is a molecule, this is a quite complex molecule, okay. Its name is OM174, and this is basically a TLR2 and TLR4 agonist. It activates the TLR2 and TLR4 mediated signaling and induce inflammation or all innate immune responses, okay. So this molecule is used in the treatment of cancer, and this is in phase 1 clinical trial. There are several molecules, as you can see in this table. These are the basically compound that target TLR for the treatment of cancer, okay. Here you can see there is a TLR3 ligand which is double stranded RNA molecule. There is a TLR2 ligand which is a autoclaved mycobacteria. Try to understand, mycobacteria, they have a variety of PAMPs, a huge, it's a heterogeneous complex of PAMP, okay. You can have the lipid and sugar complexes like lipoarabinomannan, which we call it as a LAM. There are mycolic acid and so on and so forth, okay. So it's a very good mixture of ligand which can non-specifically activate the immunity. And just for your information, here I would like to say that we use one adjuvant, we call it as a Fruent's complete adjuvant, okay, which is basically potentiate the activity of vaccine, okay. So vaccine, or if you challenging some antigen in the mice, so this Fruent adjuvant, the Fruent complete adjuvant is used only in animal or in mouse, okay. It is not used in human, please remember this thing. And that Fruent's complete adjuvant consists of heat killed mycobacteria, okay. So heat killed mycobacteria is having a tremendous potential in order to activate innate immune response, in order to make a appropriate adaptive immune response, which is a B cell mediated response, which finally result to the production of a high titer antibody. And it is also important in inducing the T cell mediated immune response. So here you can see that this autoclaved mycobacteria is also used in or about to use in human, okay, and that may be used for the treatment of cancer. There is a TLR3 ligand, which is a basically a double stranded RNA mimic. There is a TLR5 ligand, which is a flagellin protein, okay, flagellin which is present in the flagella of flagellated bacteria, okay. TLR9 ligand, which is basically consists of CPG motif of DNA. It's a basically oligonucleotide which is hypomethylated. There is no methylation, so they also have a good potential to activate the TLR9 mediated signaling. There is some non-coding stem loop DNA which is also used to activate this TLR9 mediated signaling. There are TLR7 ligand, that is single stranded RNA molecule, and there is a TLR2 and TLR4 ligand which is lipid A. If you remember the structure of lipopolysaccharide, okay, so you will find out that there is some O antigen, okay, there is a outer core and there is a inner core and there is a lipid A which is phosphorylated, okay. So this lipid A is having a potential to activate the TLR4 signaling, but it will not induce the pathogenic kind of response, it will not activate too much. So this lipid A is used as it's a quite potential molecule to activate the TLR4 mediated signaling, okay. And this can be also used as a vaccine adjuvant. Just again I am giving you one very important information. In human we use only one adjuvant which is clinically established, okay, and that is the alum. There are so many kinds of adjuvant, but in human or in human vaccine only we use the alum, okay. So you can understand we are in the need of having more vaccine adjuvant. So probably lipid A, or there is a one derivative of lipid A which we call it as a MPLA, can be used as a vaccine adjuvant. Besides this, this is used for the treatment of cancer, okay. There is a TLR9 ligand, again this is a short DNA oligonucleotide, CPG oligonucleotide. So these are basically also they have a potential to use in patient, and they are in a very good level in clinical trial. This is in phase 2 clinical trial, okay. several ligands of TLR7, which is a basically small molecule, single stranded RNA. This is basically in clinical trial, and it is in phase 2 trial. So most likely in near future we will have a kind of good repertoire of molecules which can be used for the treatment of various cancer. Okay. There is a poly-TLR. So this autoclaved mycobacteria, I told you this is a complex mixture. It can consist of all kind of molecule. So this can be also used for the treatment of cancer. Now I will move to some molecule which can be used in some infectious disease. It could be a bacterial, it could be a viral. Here you can see that there is a one molecule known as imiquimod. This is basically a TLR7 ligand, and it's an agonist. Okay, it potentiates the TLR7 mediated signaling pathway, and this is used against the papilloma virus infection. Okay, here there are some molecules. One is TAC242, which was proposed to used for or used against sepsis or shock condition. Okay, LPS mediated sepsis and shock, but somehow most likely this has some side effects, so that their development is stopped and it is discontinued. However, on another hand, there is another molecule known as eritorin. Eritorin is basically also a TLR4 antagonist, and it is in phase 3 clinical trial for sepsis. Okay. So you can understand, if you inject this molecule, then this molecule will compete with this LPS molecule, and it has a more, if it has a more affinity, it will bind to the TLR4 ligand, but it will not activate not TLR4 ligand, TLR4 receptor, and probably it will not activate the signaling. Okay, it will bind, but it will not activate the signaling. So in that way we can treat the sepsis condition. This is one way. Another way is that whatever LPS is there, you can sequester by some molecule, and then you can remove, and in that way you can treat the sepsis, LPS mediated or gram negative bacteria mediated sepsis. Okay. And over there in current scenario we use one molecule known as polymyxin B. Okay. And this polymyxin B basically is very strongly bind with the LPS. Basically we use in our laboratory in order to remove the LPS in our stimulating samples, because you know that LPS is a very strong stimulator. So if your sample is containing LPS, then it will give incorrect results. Okay. This is another molecule which is again TLR4 antagonist. It's ibudilast. Okay, and this molecule is basically used in ocular inflammation. Okay, and there is a list of molecule which is used in various viral and bacterial infection. Here you can see that TLR7, TLR9 antagonist. It is a bifunctional inhibitor. Basically this damps the TLR7 and TLR9 mediated signaling. There is a TLR9 agonist, okay, that is CpG oligonucleotide, and there are TLR7 single stranded RNA molecule, TLR5 stimulant agonist, which stimulate this TLR5 mediated signaling. It is a flagellin from Salmonella typhimurium, and there is a TLR3 ligand, that is double stranded RNA molecule, TLR7 or TLR8 agonist, that is single stranded RNA molecule, and like that there are several ligands which is used which can be used in a treatment of various viral and bacterial infection, and many of these molecules are in phase 3 trial. And very interesting, there is a TLR7 agonist which is already approved for clinical use. Okay, this is imiquimod. Okay, so Some TLR ligands are also used in non-infectious disease, that is allergy, asthma and autoimmunity disease, okay, like TLR4 antagonist antibody or TLR2 antagonist, which is a kind of most likely it is a monoclonal antibody, and it is used in the treatment of this acute and chronic inflammation, autoimmune disease, okay, ischemia, okay. So TLR2 agonist, antagonist is basically a viral derived peptide. It can be used in inflammatory bowel disease, rheumatoid arthritis. TLR7/9 antagonist, which is DNA based compound, it is used in autoimmune disease such as SLE, rheumatoid arthritis, multiple sclerosis. I have discussed multiple sclerosis earlier. TLR4 antagonist can be also used as a used in colitis. It's a colon inflammation. Okay, TLR9 CpG oligonucleotide is also used in asthma, allergy, rhinitis. Okay, it's a pollen allergy. Okay, TLR9 agonist is used in allergy and asthma. Again, TLR8 agonist is also used in allergy. So here you can see in this list of molecule, there are several molecules which can be used in the treatment of various allergy, asthma and autoimmunity disease. So overall, what I would like to say that this discovery of this pattern recognition receptor TLR is very important, and I hope in future we will see a lot of therapeutic in our clinical setup. Okay, and with this I will stop and complete the TLR, and in next session I will move to the another pattern recognition receptor, and that will be the Rigi-like receptor. Thank you.